All right, so we're gonna just do this one will be a pretty quick one. Just gonna talk a little bit about kind of the idea of the different things that need to happen in order for urine to form. Uh, when we start talking about what the kidney's doing, the real idea with the kidney is we're wanting to get rid of waste products that we don't want to keep, and then trying to maintain as many of the metabolites that are in the blood that we can. So the goal of urine production, again, like it says, there, maintain obviously homeostasis. That's the goal of most everything we've talked about. But what we're doing is really regulating how much water is in the blood, the different metabolic byproducts that are in there. Again, trying to save those things that we need to maintain and to excrete things that we do not. So the big thing is we want to get rid of a lot of these nitrogenous wastes that are present in the blood as part of our normal metabolism. Things like urea, which is part of protein breakdown, other stuff like that, creatine, from some of that muscle contraction, other stuff that we generate, as well as uric acid, which is recycling some of those nitrogenous bases. We want to be able to get rid of that stuff along the way, maintaining a lot of things that we want to keep, things like sugar and actual proteins, things along those lines. All these waste products generated by the cells end up in the interstitial fluid, sooner or later end up back in the bloodstream. So dissolved in the plasma of that bloodstream, as I said a number of times, what's in the blood? Anything that needs to get in or out of the cell. And a lot of the cases with the kidney, what we're trying to get rid of is the stuff we want to get out of the cell, and that sooner or later we want to get out of the body so we can continue to remove it from the cells. So these waste products generally are going to be concentrated about 4 times their normal plasma concentration when urine is actually finally made. We're trying to do this with minimal water loss and, again, get rid of these waste products. So this, to me, is one of those interesting slides that says a lot more than just a normal single slide does. This is kind of looking at urine concentration of certain metabolites versus plasma concentration. And to me, some of the stuff at the top here, the different ions, the thing to realize is urine volume, you're making about 1.2 liters of urine a day. Plasma volume, you have about 4 to 6 liters. So we are concentrating this somewhat even on that level when we're talking about doing that amount in there. But the main thing you see is a lot of these things, sodium, really it stays at about the same concentration. So yes, we have less than that volume, so it's not raw amounts, the same amount that's in the plasma, but you can kind of see some of these things we're regulating very tightly, other ones not as much. But again, you're going to lose some sodium in your urine. You're going to lose some potassium, actually quite concentrated relative to how much is in the plasma, where you see chloride, salt, basically sodium and chloride, pretty similar levels to what's in the plasma. Bicarbonate ion, we actually have a lot more in the bloodstream, and we tend not to lose as much of that in the urine. One of the things that is very obvious is if you start looking at urine, it's only 1.2 liters a day versus 4 to 6 liters, the concentrations are still much higher in that bigger volume of glucose, almost none in the urine. Lipids, almost none in the urine. Very little amino acids, no proteins. Those are the things that we try to make sure we don't have end up in the urine. And then the waste products, you can see urea. We're concentrating it many times over. 9 times over, 10 times over. Creatine, much more concentrated, 10 times more concentrated in the urine. You can see some ammonia, some uric acid, but again, the main thing is nitrogenous waste products, way higher levels in the urine than what we see in the plasma because we're concentrating those and getting rid of those. Metabolites, way lower levels than what we see in the plasma because we're trying to keep all those. We're going to see urine is really the process of forming is really a combination of 3 different things. We are going to make a filtrate of the blood. That happens in the glomerulus, in that glomerular capsule, using blood pressure to force some fluids out. We're going to get this filtrate, and then a lot of this is actually modifying what is in that filtrate. So filtration is the first process here. Without a filtrate, we can't do these other 2 processes. So to me, being able to form a filtrate is a pretty big deal. We then are going to reabsorb a lot of the stuff that we don't want to become part of urine, things like sugar, things like proteins, other stuff like that. Anything that ends up in that filtrate that we don't want to lose into the urine, we reabsorb it. So sugar, for example, glucose, we reabsorb all the glucose. It becomes part of the filtrate. We just reabsorb all of it back out. And again, how are these things moving? Diffusion, osmosis, active transport. We're using all these processes in order to reabsorb stuff here. moving from those tubules where the filtrate is at across those walls back to blood capillaries so we can reabsorb all those nutrients and things that we want back. Excess stuff, waste products and all that, decent amount of water remain in that filtrate, and those are what we might be excreting later on depending on other things that happen here. The other thing that we do is there is certain things that sometimes we want to get rid of that don't end up as part of that filtrate. So sometimes with like acidity or alkalinity, if we want to change that, we will excrete bicarbonate ions or excrete hydrogen ions in order to modify that blood pH. So tubular secretion, movement of the solutes, usually we're pumping this into the bloodstream, into the urine, into what will become the urine, into the filtrate. Again, moving those things into those tubules so they can be excreted. So again, this is the three, and we're going to go into a lot more detail on all parts of this, but this is the basic process of urine formation. If I gave you a question that said, what are the three processes that are involved in urine formation, it's going to be filtration, tubular reabsorption, and tubular secretion. So filtration, we are making a liquid, filtrate of the blood at that glomerulus, sending this into that glomerular capsule. Then through the rest of that nephron, we're going to reabsorb those things that we don't want to lose and get rid of those things that didn't become part of the filtrate that we want to get rid of with that tubular secretion. The main thing is, and this is the last thing we'll touch on in this one, then we'll get a lot into glomerular filtration rates and things like that on the next one here. But this membrane, it does have a number of different layers to it, which helps it to be somewhat selective in terms of what is going through this. So we have a thin, negatively charged structure, which means certain charged particles don't get through there really easy. And what we're going to see is there's these three sandwich layers that for something to become a filtrate, it has to pass through all these layers. So you have the endothelium of the glomerulus, so the blood vessel wall. It is a fenestrated type of capillary, allows plasma to travel through there and dissolve substances to pass through it. Really large things like blood cells do not pass through this. We then have the basement membrane underneath that. Glycoproteins and other stuff in there. Large plasma proteins can't get through this. Oh, I hit the wrong direction. Finally, you have the visceral layer of the glomerular capsule. So these little cells called podocytes that have these little feet that intermesh. These wrap around the capillary and don't completely sheath it, but they give you these little slits. And these are going to really stop a lot of those smaller proteins from leaving. So what we're trying to do is really be selective somewhat on what can become part of this filtrate. We do have what are called mesangial cells that are these phagocytic cells that have contractile properties that can kind of mess with this slit a little bit as well. And you can kind of see the idea of these podocytes in the three different layers here. So you can see the fenestrated capillary, the basement membrane of that, and then these actual cells, these podocytes that are wrapping around and doing these filtration slits. Again, anything that becomes part of the filtrate has to make it through all three of those layers. And again, it's keeping a lot of these blood cells, large proteins, even smaller proteins, charged ions from getting through this, but letting things like water and, like I said, small products, glucose, amino acids, certain electrolytes are all going to travel through there, along with waste products are going to get in there and become part of that filtrate. And then again, we're going to modify that filtrate based on tubular reabsorption and secretion. How much of this do we make a day? And this is what I always kind of say is we reabsorb 99% of the water that we make as a filtrate. If you were to actually somehow be like excrete all the filtrate you make, it's 180 liters a day. It's 45 gallons of filtrate. So obviously we're filtering the same fluids over and over and over again throughout the day because we only have 4 to 6 liters of blood total. So we obviously can't be making a bunch of the stuff that we're getting rid of all of it. So 180 liters of filtrate is produced a day, meaning the vast majority of all of it is reabsorbed. So this filtered plasma, like I said, we're going to have a lot of the stuff being reabsorbed. These blood components are staying in there. Blood cells, things like that. Large proteins stay within the blood. Like it says here, some stuff might get trapped and phagocytized. Again, there's going to be certain things that freely travel in there. So water, glucose, these small amino acids, these freely travel into that filtrate. There's large things that don't travel at all. Large blood proteins, as well as blood cells. They're not going to become part of this filtrate at all. They can't pass through there. Sometimes if blood pressure gets really out of whack, things like preeclampsia, you will start pushing large proteins. That can be telling you something's wrong, actually. And then there is these limited filtration things here, certain charges, proteins of intermediate size and a middle size area. Some of these might end up in that filtrate, some might not. Depending on their charge, depending on their size, they may get into that filtrate. Again, we don't lose a lot of those ones, so we are going to reabsorb a lot of that. So what we'll talk about the next time is getting into a lot more detail about how we favor this filtration, what can we do to modify the filtration rates, and things along those lines.